Chicago, a genetics clinic, 2019. A woman in her mid-40s sits across from a counselor, Italian surname, Catholic upbringing. No Jewish family history, none that anyone ever told her. She came in because her doctor flagged something. And now there's a sheet of paper on the desk between them, and the counselor is saying a phrase she has never heard in her life. Ashkenazi Jewish founder variant. The woman stares at the paper. She says, "I don't think I'm Jewish." And the counselor has no good answer for that, because the paper says what it says. What was found in her cells is called BRCA1-185 del-AG, a specific deletion at a specific position on chromosome 17. It is not random noise. It is a molecular fingerprint, one that traces directly to a medieval European Jewish community compressed catastrophically to near extinction between 700 and 900 years ago. The compression was so severe and the expansion afterwards so rapid that this single mutation now appears in roughly 1 in every 40 people of Ashkenazi Jewish descent alive today. That number stopped me cold. A 2020 study by researchers at 23andMe identified nearly 3,000 individuals carrying one or more of the 3 Ashkenazi brick founder variants. A documented fraction had no idea they had Jewish ancestry at all. They just had the mutation, moving silently through their cells, through their children, through generations of people who had stopped calling themselves Jewish long ago. Stay with me, because this next part is the reason this video exists. That mutation appeared once in a single body in medieval Europe, and then something happened to the community carrying it. Something so catastrophic, so precise, that one person's private mutation became the biological inheritance of millions. The question is not just where this population came from. The question is, what was done to them that made this mathematically possible? If you've ever actually wondered where you come from, not the family story, not the name on the passport, but the real biological answer, subscribe. Because what comes next goes deeper than most people are willing to go, and it ends in a place nobody expected. Before we get to the catastrophe, we need to understand what existed before it. Picture Erfurt, Germany, early 14th century. A Jewish quarter of stone houses, a synagogue, a ritual bath, a cemetery pressed against the city walls. A few hundred people. They speak Yiddish, trade with their neighbors, are legally classified as the property of the local bishop. They have been here since the 11th century, and by 1349, most of them will be dead. But where did they actually come from? Not Erfurt specifically, the deeper question. Where did the ancestors of the Ashkenazi Jewish population originate before they became what we now call Ashkenazi? Because the name is a clue. Ashkenaz in Hebrew means Germany. The people named for Germany, it turns out, did not genetically originate in Germany. In 2022, an international team led by Shai Carmi at the Hebrew University of Jerusalem and David Reich at Harvard Medical School extracted and sequenced ancient DNA from 33 individuals buried in Erfurt's medieval Jewish cemetery. Then they ran a statistical ancestry analysis, comparing those ancient genomes against reference populations from across Europe and the Near East. And the model that fit best? Approximately 65% of Erfurt Ashkenazi ancestry traced to a southern Italian or Sicilian source population. Not the Rhineland, not the Caucasus, not even the Levant directly, Sicily. Think about that for a second. The conventional story traces Ashkenazi Jews from the ancient Middle East northward into Germany. But the genomic data says the migration bent dramatically through Italy first, staying long enough to absorb a deep Mediterranean genetic signature before the Rhineland communities were ever founded. The Germanness of the Ashkenazim is a destination, not an origin. But here's where it gets strange, because that 65% figure is only one part of the ancestry model. There's a 19% Levantine component, a 16% Eastern European component, and a question about timing the ancient DNA has just made far more complicated. The bottleneck, the catastrophic compression that produced the mutation in that Chicago clinic, was long assumed to have been caused by the Black Death, the Crusade massacres, the documented medieval persecutions. The ancient DNA says it happened before all of them. Let me give you the number. And then I need you to hold it for a moment. 500 people. That is a medium-sized synagogue on a high holiday. A single village market on a busy afternoon. From that group, that one surviving pool, every Ashkenazi Jew alive today descends. All 10 million of them. Shai Carmi and colleagues at Columbia University modeled this in 2014, analyzing genomes from 128 Ashkenazi individuals. What they found, the entire modern Ashkenazi population traces back through a founder bottleneck of approximately 350 effective individuals, occurring no later than the 12th to 14th century. The 2022 Erfurt Ancient DNA Study refined this further, confirming the bottleneck had already occurred by the 14th century. The compression began earlier than anyone had assumed. Here's what that does to a genome. A bottleneck doesn't destroy a population, it locks it. Every genetic variant in those 350 to 500 founding individuals gets frozen in place and amplified mechanically, mathematically, with every generation of expansion that follows. Disease-causing mutations that would normally stay rare become common, not because they're useful, simply because the people carrying them survived. And survival multiplied everything they carried, including the BRCA variants from that Chicago clinic, including Tay-Sachs, including Gaucher disease. Not coincidences. The arithmetic echo of 500 people whose private mutations were passed to 10 million descendants. And then there's this. When the Erfurt team examined those 33 individuals from the 14th century cemetery, they found that more than a third of them shared a single mitochondrial lineage. Mitochondrial DNA traces only the direct maternal line, mother to daughter, unbroken. A third of the Erfurt community descended from one woman. One specific woman, living sometime before the 14th century. Her name is in no document we have found. Her grave has not been identified. But her maternal signature is still present right now in millions of living people on every inhabited continent. Most people don't know that yet, but we are about to understand exactly why it matters. Norwich, England, February 6, 1190. A medieval chronicler named Ralph de Decito recorded what happened that morning in a single sentence. Crusaders preparing to depart for Jerusalem decided, as he put it, to rise against the Jews before they invaded the Saracens. What followed in the Jewish quarter of Norwich left no monument, no memorial, no named survivors in the historical record. What it left at the bottom of an old well were 17 bodies, 6 adults, 11 children. Construction workers found them in 2004 during an excavation for a shopping center. The bones were at strange angles, some head first, the position consistent with bodies thrown, not buried. In 2022, Dr. Selina Brace of the Natural History Museum in London, working with colleagues at the Francis Crick Institute and the University of Cambridge, published the full genomic results in Current Biology. 6 of the 17 individuals had DNA preserved well enough to sequence whole genomes. The results were clear. Ashkenazi ancestry markers throughout. Disease variants genetically consistent with the Ashkenazi founder pool. Radiocarbon dating placing their deaths between 1161 and 1216. Overlapping precisely with the 1190 massacre. And the detail that stopped the research team. 3 of the 6 were sisters. They shared the same mother. Their ages, estimated from their bones, ranged from roughly 6 to 18 years old. 3 sisters at the bottom of a well, dead sometime in the late 12th century, whose genomes were fully sequenced 832 years later, and whose ancestry connects in a direct unbroken line to the same population we've been tracing, through Erfurt, through Italy, and through that genetics clinic in Chicago. And yet, the data said something nobody expected. Among the disease variants in the Norwich genomes, markers genetically consistent with Ashkenazi founder mutations. Pre-bottleneck, those specific mutations were already present in this community in the 12th century, before the worst compressions had even begun. The bottleneck did not create them. It amplified what was already there. These sisters are among the oldest individuals with sequenced Jewish genomes anywhere in the historical record. The oldest genetic archive of this community's existence is, simultaneously, the forensic record of an attempt to destroy it. 8 of the 33 Erfurt individuals, buried 130 years after Norwich, carried specific variants that exist by name in a genetic screening panel available at any clinical laboratory today. The same molecular sequence. The well in 12th century Norwich and the waiting room in 21st century Chicago contain the same molecule. Now here is where the story splits in two. And the split is the most important thing in this entire video. There are 2 types of DNA that give us 2 completely different windows into a population's past. The Y chromosome traces only the direct paternal line, father to son, unbroken across thousands of years. Mitochondrial DNA traces only the direct maternal line, mother to daughter, equally unbroken. In most populations, these 2 signals tell roughly the same geographic story. In the Ashkenazi population, they point in completely opposite directions. In 2010, Gil Atzmon and colleagues at the Albert Einstein College of Medicine in New York published a landmark genome-wide study in the American Journal of Human Genetics analyzing Jewish diaspora populations at scale. What they found on the paternal side was unambiguous. The Y chromosome haplogroups dominant in Ashkenazi men, primarily J and E1b1b, are Near Eastern signatures. Compare Ashkenazi paternal lineages against European host populations. They don't match. Compare them against other Jewish populations from the Middle East, nearly indistinguishable. The fathers of Ashkenazi Jewry came from the ancient Near East, from the Levant. from communities of the Roman period Jewish diaspora that dispersed from the region 2,000 years ago. That is a complete and satisfying answer. The fathers trace to ancient Judea. The story makes sense. Everything I just told you? Hold on to it, because it's about to mean something completely different. Because the maternal signal says something else entirely. It has always said something else, and for decades the field didn't fully reckon with what it implied. In 2013, Marta Costa and Martin Richards at the University of Huddersfield in the UK published a study in Nature Communications analyzing 956 complete mitochondrial genomes from across Europe, the Near East, and the Caucasus. They were looking specifically at the 4 major Ashkenazi mtDNA founder haplogroups. The 4 maternal lineages carried collectively by about 40% of all Ashkenazi Jews alive today. These are the founding maternal signatures of the entire population. And what the study showed was that all 4, every single major founding maternal lineage, has its deepest roots in prehistoric Europe. Not the Near East, not the Caucasus, Europe, most likely assimilated in Southern and Western Europe during the first millennium CE. When they extended the analysis to minor founder lineages, more than 80% of all Ashkenazi maternal ancestry traced the same direction. The fathers pointed east, the mothers pointed west. What this means in human terms. The men who founded early Ashkenazi communities carried Y chromosomes from the ancient Levant. But the women who became the founding mothers were local, European women who entered the Jewish community, converted, and whose descendants have been fully Jewish by every religious standard in the centuries since. This is a documented and honored tradition in Jewish history, but think about the legal structure. Judaism passes through the mother. Jewish identity is matrilineal. A population whose founding fathers were Near Eastern and whose founding mothers were European converts is a population whose entire legal chain of descent runs through women whose biological origin pointed in the opposite direction. The consensus view states that Ashkenazi Jews are a Near Eastern population with European admixture. That sentence has the subjects facing the wrong direction. The paternal line is Near Eastern. The maternal line, the one that legally defines who is Jewish, is European. And what the conversion of those founding mothers set in motion is something no one fully understood until the Erfurt cemetery gave up its teeth. Actually, hold on a second. Before we go further, I want to ask you something directly. If you did a DNA test tomorrow and it came back with ancestry that completely contradicted everything your family ever told you, would you follow that thread all the way back, or would you rather leave the story as you inherited it? Drop that in the comments. I mean it, I read them. Because the answer you give right now might look very different to you by the time this is over. I want to introduce you to someone. She has no name in any document we have found. In the research, she appears only as a designation, one of 33 individuals recovered from the medieval Jewish cemetery of Erfurt, Germany. A rescue excavation in 2013, triggered by plans to convert a 15th century granary into a parking lot, uncovered the graves beneath it. The cemetery had been sealed under that building after the Jewish community's expulsion in 1454. Her remains had been in that ground for roughly 650 years when a detached lower molar was collected and sent to a laboratory. From that single tooth, the team extracted and sequenced her complete genome. She was approximately 35 to 45 years old at death, buried in a Jewish cemetery in accordance with Jewish law. She carried the Ashkenazi mitochondrial haplogroup. She is a direct maternal descendant of the founding lineage, shared by a third of the entire Erfurt community. Her autosomal ancestry fits the Southern Italian or Sicilian source model precisely, and a pigmentation analysis of her genomic data, the same machine learning approach used in modern forensic casework, indicates she likely had light-colored eyes, possibly blue. This isn't just ancient history. This is in your DNA right now, if her maternal line is in yours. What the 2013 Costa and Richards study makes unmistakable is what she represents. Her maternal line was not brought from the Levant. It was European before it was Jewish. Some woman back in her direct maternal chain entered the Jewish community, converted, raised children in it, passed her mitochondrial sequence forward through every generation that followed. Her descendants passed down the prayers and the calendar and the law, and also her European maternal signature, to people living right now. We know her approximate age, her likely eye color, her ancestry proportions, 8 disease variants she carried that a doctor can screen for today. And we know the bottleneck had already occurred by the time she died. Her community already the compressed remnant of something larger before the centuries of persecution closed in completely. She is not a paradox. She is what the founding generation actually looked like. And here is what the Erfurt research revealed that no historical document had preserved. Medieval Ashkenazi Jews were not one unified genetic population. They were two. The study identified two distinct subgroups in that cemetery. One with higher Middle Eastern and Southern European ancestry, genetically close to modern Ashkenazi Jews from France and Germany, and a second with a substantially higher Eastern European component, suggesting connections to Jewish communities further east. Two groups, same cemetery, same language, same religion, separated by a genomic difference invisible to everyone alive at the time, detectable only from sequenced teeth pulled from parking garage soil in 2013. By the time their descendants reached the modern era, that internal distinction had vanished completely, compressed into a single, nearly homogeneous genetic profile shared by Ashkenazi Jews across every continent on Earth. What did the compressing? You already know part of the answer, but not all of it. March 21st, 1349. Erfurt. The Jewish community here had existed since the late 11th century, one of the largest in Germany at its peak. On that day, during the Black Death, accusations of well poisoning swept the city. Several hundred members of the community were killed, the survivors expelled, a granary built over their cemetery. 5.5 centuries later, a parking structure project cracked open the soil above their bones. That is one event, one city, one date. Count the others. The Rhineland massacres of 1096. Crusaders moving through the Rhine Valley, destroying Jewish communities in Worms, Mainz, and Cologne before they ever reached the Holy Land. The expulsion from England in 1290. The expulsion from France in 1306. The wave of 1349 massacres across Central Europe. The expulsion from Erfurt itself in 1454. Count them backward from today. 72 years, 135 years, 282 years, 330 years, 930 years. Each one a compression. Each one removing people and their genetic variants from the pool permanently. Each one narrowing something that could not be widened again. Here is what the Waldman et al. Study confirmed about the genomic result. Today, an Ashkenazi Jew from New York and an Ashkenazi Jew from Tel Aviv, two people whose families may have been on different continents for a century, whose languages and cultures diverge significantly, are genetically nearly indistinguishable. David Reich of Harvard stated it plainly in the research. Jews in Europe were a religious minority that was socially segregated, and they experienced periodic persecution. The data confirms exactly what that means at the molecular level. That genetic sameness is not a mark of cultural solidarity. It is the shape of everything that was removed. Geneticists measure something called runs of homozygosity, long, identical stretches of DNA shared between unrelated individuals. The longer those stretches, the more recently those individuals shared a common ancestor. Ashkenazi haplotype blocks are longer than those of any comparably sized European population. The proximity is the mathematical signature of compounded mass death across centuries, driving the same descendants closer and closer together with every generation. The two medieval subgroups from Erfurt, the Middle Eastern-leaning group and the Eastern European-leaning group, merged into a single compressed profile not through peaceful integration, but through the removal of everyone and everything that did not survive. And what survived carries a medical price that is still being paid today. And this connects directly to what a lot of you raised after the Yamnaya video, the question of whether population genetics changes what it means to belong to a civilization. Because here's the version of that question the Ashkenazi data forces you to sit with. If the founding mothers were European women who converted, and if every traditional legal definition of Jewish identity has run through the maternal line ever since, what does origin actually mean? Is it where the fathers came from, or where the mothers did? Drop that in the comments. I genuinely want to know where you land on it. We are back in that Chicago genetics clinic. 2019. The same woman, the same paper on the desk. The three Ashkenazi Brisa founder mutations, known clinically as 185 del-Eg, 5382 NC, and 6174 del-T, are present collectively in approximately 1 in 40 people of Ashkenazi Jewish descent. In the general population, the equivalent rate is 1 in 400, 10 times more common. That elevated frequency is a direct consequence of the bottleneck. Those mutations were present in the founding population of 350 to 500 people. As that population expanded to 10 million, they expanded with it, not because they were advantageous, not because selection chose them, but because the bottleneck had removed so much surrounding genetic variation that these private variants became proportionally dominant in every generation that followed, 1 in 40, from a compression before the 14th century, still encoded in living cells today. And here is the part that changes how you read everything you've seen in this video. A 2020 study by researchers at 23andMe confirmed that a significant fraction of carriers of these Ashkenazi founder mutations did not identify as Jewish. The mutation does not require the identity to survive. It only requires the genome. passing through bodies that intermarried, emigrated, assimilated, forgot, invisible, generation after generation, until a clinic would print out and says a phrase someone has never heard before. The tooth pulled from Erfurt soil in 2013 contained a pathogenic variant that an oncologist in 2024 can screen for with a single blood draw. 700 years, the same molecule, passed without one generation missing, through expulsions, the Atlantic crossing, changed surnames, Catholic baptismal records, arriving in Chicago still carrying the molecular signature of the community that first compressed it into common currency. Mutation is not a defect, it is a testimony. And then there is a question researchers are now beginning to ask carefully, because the evidence is early and contested. Could the biological consequences of multi-generational persecution extend beyond founder mutations into epigenetic changes, the chemical marks that control gene expression, potentially passed from parents to children? In 2016, Rachel Yehuda's team at Mount Sinai published findings in biological psychiatry pointing toward exactly that in Holocaust survivors and their offspring. The field hasn't settled it. The research is still running. What it finds next, nobody can say yet. Before I give you the final piece, I need to make it personal. If you have any Ashkenazi ancestry, confirmed, suspected, or possible through an Italian or Eastern European family line you haven't traced, there is a clinical probability you carry one of these founder variants. The 1 in 40 figure is real. The screening exists and is available. And for a significant fraction of the people who carry these mutations, no one has ever told them they should ask. The woman in the Chicago clinic didn't know to ask. Her family had lost the thread generations back. What the genetics found was older than any living memory, tracing to a community compressed before any record her family kept could have captured it. She is carrying a molecular testimony from the woman in the Erfurt cemetery. The woman whose tooth was pulled from parking garage soil, whose mitochondrial signature moved through 25 unbroken generations, through conversion, emigration, changed surnames, the Atlantic, all of it, and arrived on a sheet of paper in Chicago in 2019 that said, Ashkenazi Jewish founder variant. 3 women, different centuries, different cities, no shared names. The 3 sisters at the bottom of a Norwich well, dead in 1190, genomes read in 2022. The woman in Erfurt, buried in the 14th century, a tooth pulled 650 years later, and the woman in Chicago in 2019, holding a paper that connected her to both of them without a word of explanation. They are not 3 separate stories. They are one, running in a single unbroken molecular thread from a medieval community compressed almost to nothing, through expulsion and massacre and demographic collapse, and arriving intact in living bodies today. The thread did not break when the communities were driven out. It did not break when the names were changed. It did not break when the ocean was crossed. If you found this video because of a DNA test that told you something you didn't expect, you are not at the end of this story. You are inside it. The genome does not mourn. It only remembers, and it does not ask permission to remember in you.